our kind of okay, yeah. So, and I'm really delighted to share kind of our vision. So what David said about like reproducibility, data science, and I think benchmarking fits into that. So I think, so I will spend substantial part of the talk talking about that, but I think also I will share our vision for kind of cultivating computational culture, and I think a lot of kind of in common, or like we can exchange ideas from that node, and then we'll talk about what we do in the School of Pharmacy as I think as the perfect example. So there is a school which lacks computational culture at all, and if we, given a chance to build a computational culture from scratch, like what do we need to do? So this is very active discussion we have in the School of Pharmacy. I will share my vision, and I hope some of that overlap with the entire school vision. I think it's okay. Okay, now this works. Okay, great. So I want to start and say that I hope the message, which I guess will get some support in this audience. So we'll say that the data take the central stage across the domains. I think for us, it's biological science and then genetics and other fields, but I think across the fields, right? The data enables the stuff we were not able to do before, and having access to the data, we can answer important questions computationally. I think that's actually quite, if you kind of think about that the computational researchers are well positioned to make as significant discoveries as the people generating the data in the wet lab. So that's quite substantial to me. So there is a lot of room where bioinformatics / computational algorithms, statistical algorithms can enable novel discovery, can find biomarkers, and pave the road to the precision medicine and genomic-based medicine. There are a lot of barriers which needs to be addressed. I think the data comes nicely, but it doesn't come perfect, right? We have a lot of shortcomings of the data, and they need to be handled and leveraged before we talk about integrating the genomics and bioinformatics in the clinical decision making. So what are those barriers? I guess the errors, right? We're all aware of the errors which data has, and they can come from the sequencing technology, like sequencing error, or they can be human errors, right? Like the shortcomings in the data, and as well as the throughput, right? It's like there is so much data that we need effective algorithms, and I think this is how my lab, and I imagine many people here, this is how we see our contribution to the field, that we enable the effective and accurate data processing by developing effective and accurate algorithms. And this is how I see bioinformatics, at least that's my vision, right? So this is the frontier between noisy and large data sets on the left and meaningful interpretation, which physician, clinician, and life scientist can use, right, on the right. So today I will talk about one aspect of the research we do. It's about benchmarking. So this is something, I guess, quite an interesting topic when we started that. So we wrote that review a while ago when I was a postdoc at UCLA, and I think there is still a lot of kind of mixed opinions about benchmarking, right? Like, is it real science, right? Like, is it real contribution to the science, and how that should it be published at all? This is the feedback we were receiving as we pursue that road of doing benchmarking of existing tools in what we think is in the most effective way. So we get a lot of feedback. Is like, oh, is it real science? Is it like just engineering project? And like, should you be published at all? So there is, I guess, a mixed opinion. Some people worry about that to be negative, right? That's kind of cultivates negative sentiments in the community, meaning that you evaluate others' work. And some worry that, that not all the methods are the best. And I guess that's, like something that when we thought about benchmarking, we realized that nobody kind of talks systematically about benchmarking, in the sense like, what is that process and how it should be and why we are doing that? What's the value and what are the best practices? And I will present some data which we looked at 20 plus existing benchmarkings. And I think, in my opinion, we are far away from doing best practices, even where we can. So sometimes it's hard because it requires additional budget, access to the specific wet lab experiments to make comprehensive gold standard. But sometimes it's just the culture. It's a pure culture. Like, you can do it, but you're not aware of that. Maybe you don't care enough. So I think there is a lot of room to improve in both ways. So I think benchmarking answers, if you want to kind of start very high level, I think benchmarking answers the question how to choose the best tool, right? And the tools are different, they perform differently in the different settings, but how do you choose the best one? And I guess the problem or the issue which we're facing, can the can every single tool be the best, right? But this is the impression you're getting if you read individual papers, right? You take the paper and perhaps the reason that paper is published or the method is published because the method is the best. But can all the methods be the best, right? And here we're talking about what is called self-assessment trap, right? So we perhaps, when we consciously or maybe unconsciously, right, when we evaluate the tools, we are falling in the trap. So then, to avoid that, we would like to establish kind of independent benchmarking where the whole task, because if you think about individual methods, right, your goal is to develop the method, but then you need to self-assess, right? You're stretching thin, you're developing the method and assessing that and so on. As benchmarking, you kind of step away and say, hey, I just want to assess how accurate the tools are, how robust they are, how scalable they are, in the most independent way, using the most comprehensive data I can get access to, and then do it rigorously, right? Like as much as I can, and kind of don't worry about like particular method, who developed that method, and so on. So that's kind of the ideal scenario. So on the very kind of simplified way, you know, like you can think about that you have software tools you want to assess, and you know, like they do some performance according to comparing to the gold standard, right? So you have a gold standard, like the truth. So the reason we don't say truth, so it's not all the time the truth, right? So sometimes it's impossible to get the truth, and we kind of use the proxy of the truth, which is gold standard. In other words, the most accurate way we can measure a particular thing. And actually, it's interesting to realize that for some problem, we will never get the ground truth. So think about read alignment, right? Will we ever be possible to get true alignment of the reads? I would argue that never, because it's an artificial problem, right? It's not biology. There is no biology in the read alignment. We take somebody else's DNA and we match the reads, my reads. So individually through that DNA, there's no biology, right? It's just like a trick we do to enable discovery of the variants and so on. So there is no way, at least I'm not aware of way, except simulations, but in the real way to know the true position of every read, right? So it's limited, right? So it's not ground truth, but the gold standard. So we have the gold standard, we have some scoring metrics, right? Like the way we want to compare the gold standard and the prediction from the tools. We have some raw data sets that can be, let's say, some omics technologies, like some genomics data and ASIC data, maybe whole genome data, and so on. And we have software too, right? So this is what we're assessing. We want to assess the accuracy of those tools, and they have some different kind of distribution of scoring, and we select perhaps the best tool under different conditions and under different metrics and so on. So that's kind of the simplified way of, the very high level way to do benchmarking. So in terms of gold standard, I think that's absolutely crucial part of the benchmarking, right? So I think the way you can think about benchmarking, if you want to perform the benchmarking, do you have access to the accurate gold standard, comprehensive gold standard? If yes, that's great. Perhaps you will be able to get to do a comprehensive benchmarking, and sometimes again you don't have, and sometimes you just do have access to the simulation data and like a read alignment, and probably that's fine, even though there are shortcomings of the simulation data. And so here we try to classify every way we are aware of you can generate the gold standard, right? So often people use alternative technology, right, to generate gold standard. It's not necessarily more accurate, but at least orthogonal, right? So give you some good sense. Some trusted technology like Sanger sequencing, right? So we know that it doesn't produce the errors. So if you want to check the variants in the particular region, perhaps, and you don't need the whole genome and so on. So you can use some trusted technology, which you absolutely trust. Often people use multiple ordinary technologies. So the example of that, for example, is genome in the bottle, right? Where people took, I think, trio, and then they run all available sequencing technology, PacBio and Nanopore and Illumina, and then basically they look at the overlap of those and they define the true variants, true structural variants and true single nucleotide variants. And that's a great way to do it. And again, every such technology comes with the limitation. In this case, right, so you rely on the overlap, but the overlap doesn't necessarily cover the entire genome. So you're talking about the subset of the gold standard rather than the complete one. Computational simulation, I think we all use that a lot, and that's excellent because it comes with the free of charge. except the cluster time, right? And that's great. I think that's a great start. And especially it's great because you can alter any possible kind of technological or clinical parameter, right? So you can really explore the whole kind of space of those parameters. And because again, it's scalable, there is no cost. Curated software input, you can curate kind of manually or curate a database, you can create it manually or use expert manual evaluation, right? So experts are looking at the images of the cell types and they say something which we think is correct. And the last one is mock community, where basically you engineer your sample, right? If you think about the microbiome, you combine certain strains or certain species together, and you know the truth because you were the one mixing them in the certain relative abundance proportions. The limitation of that, probably you oversimplify reality, right? Because you cannot mix the true or the expected number of species which you find in the sample, microbiome sample. So you rather mix 10 or 20 or that order of species. And what you can see also, there is some part where there is a processing, like kind of computational processing required, like here, and sometimes it's just gone. It's just like direct. If you design more community, you just design that you know the truth, so you don't need to process. And this is kind of the ways you get the gold standard. So we looked across some 10 relevant peer review, what was 25. So we look across 25 studies, benchmarking studies at the time. And so what we see that there is quite a distribution of how people generate the gold standard. The most popular is simulation and alternative technology. Those two were popular, and then some others are also represented. So, and I guess, as I mentioned, each technology comes with the advantage and limitation. So if you, for example, look at the alternative technology, it's nice, it's direct, usually no computational inference is required, right? But we don't know if it's necessarily more accurate. Computation simulations, right, comes no cost, it is scalable, but probably cannot capture the true complexity of the real biological data. Okay, so people thought, I guess, a lot about how we can improve the simulation data, right? And then there are a few kind of ways we can do that. We can take real data, incorporate real, kind of combine real and simulation data in one comprehensive data set, right? So the way usually people do that, they take real reads, right? And they add on the top the variance, or they add on the top the structural variance and things like that, right? So you keep the real reads. So the distribution of the reads, the distribution of sequencing errors, things like that, they're real. However, you inserting those, let's say, variants or structural variants, so those are simulated, right? They're artificial, but you know the gold standard, you know the truth, right? You can also subsample real data set to generate new data set with the known properties, and this is what we actually are trying to do. So we're trying to design a tool which will not introduce anything artificial per se in the data, but rather will change the data, right? So the example of that would be the microbiome sample, right? The metagenomic sample, where let's say it's a more community where you know every single strain and you have the reads. So if you can assign the reads on the strains, right? So you can then change the relative abundance of those strains, right? You can just really remove certain reads from certain strain, and you can make the abundance of the strain lower, which will be more challenging to the bioinformatics tool. As well as you can actually reduce the error. Adding the error is obvious, right? You just sprinkle the errors across your data set, so that's easy. But how would you reduce the number of errors? So if you assume that you know all the strains and you have mapping of the reads on those strains, should be no mismatches, right? As soon as the mapping is correct. So actually every mismatch you can treat as error and start removing them, right? Reducing them. And then there are a few other ways we can kind of change the data, but again, highlighting, you will not introduce anything kind of artificial in the data, we just change the properties. So, and I think the idea here that we want to increase the goal is here by developing this tool, which is in process. So we want to increase the diversity of the benchmarks, right? So very few studies explore the effect of the read length, the effect of the coverage, and other things, right? Because it's expensive to generate. If you want to explore the read length effect, you need to generate individual data sets with the different read lengths, which is expensive. But if you can basically computationally truncate the reads, right? So you get some kind of compromise. So you can explore the effect of the technological / sequencing or some actually clinical parameters on the bioinformatics results. So another way is like another interesting aspect of the benchmarking, how you do that, right? Do you do that individually in the lab, or do you do what is called competition-based, right? Do you announce kind of a challenge, right? Where the developers of the tools are running the data sets on their side and give you the results. There are advantages and disadvantages, right? It's actually, so it's the person who runs quite many benchmarking, despite the process being extremely straightforward, right? I can explain the project perhaps in 10 minutes, right? Say this is the gold standard, this is the tools, this is how we compare, but there is so many technical challenges, right? The data is large, as we all aware, not all the tools are easy to run. There are issues, parameter optimization, if you want to do that, right? Increase the space of the runs you need to perform. So it's hard, right? It's just laborious, right? And so instead, if you do challenge-based, all you need to do, you need to generate the gold standard and hide it to yourself, right? And give the raw data to the community, hoping the developers of the tool will do the best job possible to run the tools. And they send you the results and you do the evaluation. So if you want, to see, so what is the more prevalent? So actually, performed by a single research group is actually the most popular way to do that, and challenge-based studies accounts for 28%, and 72% goes to individual, the benchmarking performed in the individual groups. Sometimes it's good. Sorry. I need to click twice. Okay, so another advantage actually of the challenge-based benchmarking, that you're gonna benchmark more tools, and the reason is obvious, right? Because it's easier, right? Like the people who develop the tool, they know how to run them. As you run individuals, you kind of, as you can see, start with the low number, right? You have less than 10 on median tools being benchmarked, right? So, and I guess the reason is just because it's hard, right? Many tools are very hard / impossible to install and run, and that limits the number. So actually, so based on our previous results, we observed that quite a large substantial number of tools have a complicated installation process. I think the way we define it, I think it requires more than 2 hours of installing. And actually, see if I have it here. Yeah, like many tools like comprehensive documentation or incomplete documentation, meaning that if you just blindly follow the documentation, you will be not able to install the tool. Rather, you need to seek advice from Google, or nowadays, I guess, maybe ChatGPT, what additional commands do you need to run. So it's not complete, right? And then I think we have shown that it increases exponentially the time you need, right? If you blindly follow the installation instruction, the time is nothing, order of 10 minutes or so. But as soon as you seek help, or you need to figure out yourself additional command, the time goes quite high. So parameter optimization, this is something required, and I think very essential. Basically want to find the combination of the parameter which provides the best results. And actually, many benchmarking studies found the better combination of the parameters comparable to the default ones, due to the access of the better gold standard, I imagine, or maybe checking more combinations, right? But it's very laborious but necessary task, but it requires substantial work and a substantial number of commands to be run. Okay, so another thing was, so when I mentioned about the culture, right? So there is some fundamental, I guess, barriers, right? In the benchmarking, as I mentioned, for the read mapping, right? So we are not able to find the true or molecularly defined gold standard. And sometimes it's just very expensive, right? You don't have access to the wet lab, and sometimes it's just too expensive or impossible to generate gold standard. But there are things that we can do and we don't, right? And this is what was the most surprising thing, at least to me personally, when we have found out that it's about the culture, right? So 40% of the studies don't share the code and data behind the benchmark, right? So that's something, it's kind of about the culture. I guess the people who perform the benchmarking are not necessarily aware of the value of that, or that they have some technical issues, which I don't imagine they are of that caliber. Because if you perform the benchmarking, perhaps you have some quite sophisticated technical background, because like it's a very technical project. So as it comes to data and code sharing, I guess it's less technically challenging. I guess there are a few factors contributing to that. I think we don't have, especially for the code. For the data, we have a bit better policies, but for the code, it's almost like Wild West. There is almost no requirements, or at least many journals don't have any requirements as it comes to the public sharing of the code. I think infrastructural challenges, and as soon as the data is simulated or somehow perturbated using any computational approaches, it becomes not acceptable to any genomic repositories like SRA and the GEO, and basically you cannot store it on the GitHub. So you need to find your way, like Figshare perhaps, or some other repositories, how you store that. So that can be the challenge. That was another surprise to me, that as we evaluate the tools, I imagine the accuracy of the tool is as important as the scalability, right? Like, can we apply the tool across larger number of samples? And I think it's determined how fast the tool is running and how much resources it requires. So we were quite surprised to see that 72% of the studies, they do not report CPU time or RAM in the benchmarkings, right? So that's again something, it's a matter of one command, right? And you don't perhaps need to do it on the whole data set. You just need to select some certain data sets from your benchmarking and run and assess. And yet people don't do that. So in terms of like the statistical measurements, sensitivity and PPV were the most popular measures. But again, it's very kind of laborious and delicate process how you define the measurement, because you don't want to be biased, you don't want to give preference to any tool. So that's preferably to spend quite a bit of time and to do a lot of brainstorming. How would you select the most comprehensive tool, so then you don't get any ambiguous results, or you don't bias the performance of any tool. There are a few open questions, I guess, in the benchmarking. So how we do continuous benchmarking? And because what's happening, you do benchmarking, you assess the current tools, you publish the paper, and perhaps you stop, right? And I guess it's similar. We just talked with David, like in terms of software development, right? So that's another kind of similar challenge in academia, right? So we either get some funding to develop some tool, or get the PhD student to develop a tool, or whoever is doing that. But then as we publish the paper, we see that as the end of the process, but perhaps new tools appear, new gold standards appear. How you do it, how you incentivize continuous benchmark, right? Who will pay for that? I guess that's a separate, I guess, question, right? How we get the funding. There is no currently the funding to do benchmarking. So we rely on people will and kind of interest of the people who believe that's very important to do, to continue doing that. How we do community adoption, I think the good example is the tool Top Hat for the RNA-seq alignment. I think it was shown by quite many benchmarking studies that it's suboptimal, right, to align the reads. And actually the developers of the tools, I guess, on the Twitter, on other platform, were indicating that please, use alternative tools because there are better tools. Yet the process is, I guess, it's not flexible, right? That many publications every year are still using Top Hat, right? So one reason is because they did analysis quite long ago, so they cannot change. But another one, I guess there is no culture of relying on the benchmarking, broadly speaking. Instead, I think in life science, or very often people ask each other, which tool did you use? And they trust kind of the neighbor. So it happens. So as soon as there is a critical mass of people using a particular tool, it's very hard to change that culture, right? It's very hard to change that choice. I guess another question, which I didn't put here, like the financial aspect, as well as like the how your benchmarking can receive. I think this is the comments, again, we were receiving for the benchmarking we perform, but also like when we wrote that review, right? And some people are worried about the negative kind of sentiments because it's a delicate process when you say some tools are suboptimal and some tools are better. Basically, you rank, right? It's about ranking. And it's some people take it perhaps personally. So that's another challenge, right? Like, and some people, we got feedback from many researchers, they're just worried to do benchmarking just to avoid the kind of, not the conflict, but that kind of tension, right? When you maybe see your colleagues, you kind of, say that the tool they develop are not optimal in this particular settings, and then they're worried that will be negative consequence for their career and so on. So there is that aspect. So we try to kind of formulate some very simple principle for the systematic benchmarking, at least something that we can do. Right, so I think we all can spend some time and do comprehensive list of the tools to be benchmarked, right? So we can spend time doing literature review and get the list of the tools. Prepare and describe benchmarking data in the details, and obviously share the data and use the most comprehensive benchmarking which is available to us. Carefully select evaluation metrics and pack in the form of the script, which community can use later, right? So it's not about only, so that I think one thing to realize when you do benchmarking, there is several kind of spin-offs of the benchmarking, if you wish, like one, of course, you rank the tool, that's kind of primarily thing, but then your data, like your gold standard data you generated is extremely valuable for the future benchmarking or for some other purposes. The statistical measurements you selected, so perhaps you spent a lot of time, and the metrics of those are helpful because they can be adopted in the community. And then practically speaking, kind of like the scripts you wrote for those evaluation metrics can be adopted in the community. And again, that's kind of comes to the standardization, right? Like of how we evaluate things. So I think benchmarking of those kind of flagships, which can kind of, which can start the change and promote the best practices. I think you would need to consider parameter optimization if it's not very appealing, because you need to run so many job, the processes and different combination of the optimization, but still that's necessary. I guess you would need to summarize the algorithms, so explain which every algorithm behind the tool you're benchmarking, what they're doing in that algorithm, and share the commands, how exactly did you install the tool and how exactly did you run the tool. And perhaps you want to go all the way and do virtual machine or maybe images or containers, something portable, reusable, and so on. And yeah, so if the data is not stored on this kind of standard repositories, like sequencing grid archive or the geo, if you're doing more custom repository to do that, would be preferable to have the flexible interface, so to kind of incentivize the community to download the data and reuse it. I guess there are many open questions. I would encourage the researchers community to work on the benchmarking. So how we address that the benchmarking may foster negatively competitive sentiments in the research community, and how we can find the funding, right? So those are the questions that get yet to be addressed. So more recently, we also look at the single cell data, and we looked with Mark Robinson from University of Zurich. So we look at the 62 single cell method benchmarks, and the paper is available on the preprint. I think it will be soon published in GigaScience. So we looked at how many methods people have benchmarked, how many matrices they use, how many data sets, and we also look at other kind of neutrality, right? And by neutrality, we mean other people who develop the tools are also part of the benchmarking. And I think it's a delicate question, right? So I think that's okay if they are part of that, right? But there is a danger, right, of neutrality. And it's actually surprisingly often the case that the people are, it happens, but I say not often, but it happens that the people who are doing benchmarking would actually develop the tool. I think the reason is very simple, because they do care, right? So they care about the field and they care about how the tool performs and so on. So So that being said, so I think I want to transition and present one particular benchmarking we are doing in the lab. I think it's almost complete. And then, so we'll talk about that just to illustrate one benchmarking, and then I will transition to something about teaching initiatives at the School of Pharmacy. So I hope to generate some discussion and get feedback from you guys in that. So yes, I would like to talk about the rigorous benchmarking of the T-cell repertoire profiling method for the cancer RNA sequencing data. So TCR-seq is a great technology, right? It provides the estimate of the TCR repertoire to measure the effectiveness of the immunotherapy, of overall prognosis, but it's not scalable, right? Meaning that we have limited number of TCR-seq samples comparable to RNA-seq, and still, if you go to the clinics or if you go to the cancer studies, they do RNA-seq most often, and they rarely have the budget left to do TCRs. So the question is how well RNA-seq can capture TCR-seq repertoire, and is it anyhow similar to TCR-seq, right? Under which the question was, we formulated under which condition you can use RNA-seq technology to capture TCR-seq, which will be informative and accurate. And we have some methods. So actually one of them is ours. So I think in this case, based on our kind of scale, we are not neutral. So we benchmark among 4 tools our own method as well. Okay, so some very short background. So if you think about the adaptive repertoire, that's encoded by the TCR and BCR loci. And if you look at one of loci, you have a collection of the V genes, D genes, and the J genes, which are recombined in the random fashion. But if you think about the recombination, perhaps that's not enough. Having that mechanism would be not enough to achieve astonishing diversity that TCR-seq repertoire it has. So it's around 10 into the power 15 distinct sequences, right? So what is also allowed to insert random characters between the V and G and D and J, so called non-template insertion. So that mechanism literally can generate you in a sequence. So several definitions. We'll be talking about the CDR3, complementary determining region number 3, which we'll be using as the proxy, which is the most diverse region, by the way, but also we're going to use this as the proxy to define the clonotype. And the collection of the clonotype of the particular individual we refer as the repertoire. So in terms of the short reads, if you think about RNA-seq reads, how would they map that, right? So first of all, why would you have that, right? If you're running RNA-seq experiment, why would you have TCR reads in the sample? Just because the T cells can be there, right? If the T cells are in the samples, they have their own receptor genomes, right? So they have their own genome, so something like that. So you would have some reads coming from that region. So the goal is actually to identify them. There will be among unmapped reads, meaning the reads which are discarded, right? So they didn't make it. So the reason reads which are fully mapped to the V genes are part of the mapped reads because the V gene is actually part of the genome.
Will they have the poly A tails?
Mm-hmm.
Oh, okay.
Yeah, so they will be enriched.
Great.
Yeah. So actually, if you run RIBA 0, you have almost none because there is, the selection. But if you run poly selection RNA-seq, you will enrich for them. So will be sometimes, if there is decent number of T cells, you will have some decent fraction of them. Okay, so the goal is to leverage those reads which are mapped, right? And so look among unmapped reads and find the reads which are spanning that recombination, and from that information infer the CDR3 sequence. So that's the goal of the profiling. Okay, so we will have 2 type of repertoires. One we will call monoclonal and another polyclonal. So very simple monoclonal if you have a particular clonotype which is very dominant, and some other ones, and then the polyclonal basically will have all kind of different clonotype more or less present in with similar proportions. So we'll have kind of both types of those repertoires in our data. So we try to, talking about the gold standard, right? So we try to collect all the data we can. And this is what was our contribution. So this is collaboration with UCLA, with Antoni Ribas, who kindly provided us the data. And we got 14 samples, as well as we use the public data. Anything we can find. And this is, I guess, TCGA data. And this is the previous benchmarking study. And as you can see, there's a variety of sequencing parameters as well as the throughput. But I think what is important is that some samples like PBMC and they're rich, like T-cell rich tissues, and then the skin samples will be T-cell poor tissues. So we have combination of parameters, combination of tissue types being T-cell poor and T-cell rich. Actually, I think I have it here, right? So we have 4 possible ways, right? So we have T-cell monoclonal, T-cell polyclonal, T-cell poor monoclonal, and so on, right? So we think that we cover all kind of major scenarios. And the question is in which settings, right, RNA-seq can be a good idea, a good technology to substitute or at least achieve the level of accuracy of the TCR-seq. So in terms of the method, we got 4 methods. Actually, one line is missing somehow, but we got 4 methods. So one of them is ours, IMRAP, developed by us, and another 2 methods. They change. They are different in terms of the protocol they use or the algorithm they use. Some of them, like us, we don't require alignment. Some of them do require alignment. So there's a lot of kind of differences, and they're more or less all recent. So it's a recent field. So a few results. I guess the first question we ask: what is the fraction of the repertoire we are able to capture? And when we say capture, we mean on the top. What is the total abundance, relative abundance of all the clonotypes we capture? So if you think about low SDA, low Shannon entropy, low Shannon, which is basically the monoclonal repertoire. So it's enough to capture the major repertoire, which is around 90%, and then you capture the majority based on the frequency, right? So then the results using the blue, they're quite nice, right? So we are able to capture all the clonotypes which are above 0.0003%, right? So we only kind of miss very little. As soon as the tissue become poor, meaning there is not a lot of T cells, meaning there is not a lot of reads, things are much harder, but we're still doing a good job, right? We were able to capture all the clonotypes above 0.02%, right? So things are decent, right? So we're capturing the major clonotypes, and perhaps those are the most relevant to try to predict the immunotherapy results, the prognosis of the cancer and the outcomes, and so on. So as we talk about the polyclone, things are much harder, right? And here we have decent results as we talk about T-cell rich, polyclonal in the green, but probably not that great T-cell poor polyclonal. In terms of the estimation of the diversity, clonal diversity, which is often the very popular measurement, and it was shown to be associated with the immunotherapy result, the prognosis, how diverse is the repertoire. So I think the RNA-seq doing fine, right? Especially in the monoclonal ones, right? And there is some differences between the tools, right? So you can see the mixer is probably doing the best job, and other tools are more or less similar. But as soon as we do polyclonal, things start shifting a little bit away. So the conclusion for this part is, so we say, based on the benchmarking, that RNA-seq-based TCR-seq profiling methods are able to capture the clonotypes. And actually, forgot to mention, we were talking here about the beta chain. This is the data we have got. I guess it can estimate the diversity of the repertoire in monoclonal repertoires, right? So as it comes to polyclonal, I guess that's not that accurate. And, so basically, that's just a nice way to say, just don't, if you have T-cell poor polyclonal sample, I guess then just don't do it, right? The results will be not good at all. I think the nice thing about if you use RNA-seq, first of all, the data is usually available, right? So we talk about some clinical study where they do RNA-seq to study gene expression, so it's available. So you can use only the fraction of the data where you feel confident and use it, right? And perhaps it will be still decent sample size. But what is nice, you can also do both alpha and beta chains, because usually when you do targeted TCR-seq data, you can only do the beta chain. So you can do that simultaneously. So overall, we conclude that RNA-seq is a nice approach, but you should be careful what is the tissue type you have and what to use. Okay, so So that was illustration of benchmarking. So I will use the last, I guess, 10 minutes to be on time. And so, yeah, so talk about, and actually, yeah, so before that, that's the benchmarking we're running in different stages. Some of them half done, some we just start. So if anything interesting to you, like join our efforts, we will try to benchmark HLA typing algorithm from RNA-seq data. We jumped on the wastewater, how accurate we can assemble and quantify the variance from the wastewater. Extremely challenging. It's a beautiful data. It's basically a mess in the sense of sequencing, like errors all over, gaps all over. So I think we need good bioinformatics here. We look at the databases and we try to use sequencing data to complement the databases. And the last one, we actually were surprised there is no benchmarking how accurate we can infer ancestry from RNA-seq data. So I think perhaps we can, but there is no formal kind of accuracy. Okay, so let me spend 5-10 minutes talking about how we deal with computational culture, how we pioneer or cultivate the computational culture at the School of Pharmacy. And I guess some discussion might be relevant for those of you who are involved in the teaching initiatives. So current problem is that there is no, if you think about the Council for the Pharmacy Education, so there's no such thing as like data science or code-based solutions, anything like that, right? Any of that sort. That's not part of it, right? So we want to bring that into discussion and address it. And from my perspective, there's a digital divide in pharmacy. I guess it's similar in biology, but more profound. There is a need to work with the large data. The data is there, and there is many interesting questions and insights which can be obtained from the data, but yet students have no skills, right, or limited skills to work with the data. And actually, students do want. So based on the recent paper our dean wrote, there is actually the demand. So when you ask students and the community, would you like to operate data analytics, they say yes. At least some fraction, 1/4 of them say, yeah, that's something desirable, but yet we don't provide that. And I guess there are a few limitations of that, right? I guess, in my opinion, then we need a special approach, right? So we are talking about the students who has zero, and when I say zero, I literally mean zero background in computer science. And they actually don't have desire to go too far in that domain, right? They don't want to become computer scientists, they don't want to become coding person, so on, but they want to have a skills, right? So currently there is no code-based training, hands-on training on the code-based data analytics and reproducible data science. And here maybe I'm presenting not that popular opinion, but I strongly believe that we should cultivate the code-based data analytics and instead of kind of graphical interfaces and things like that. And I think it's doable. So based on my, I guess, 10 plus, I guess already years of experience teaching that, I think if we do it right, so people with zero background can write decent code, small code and so on. So I think that gives them correct impression of the data. And I kind of, my worry all the time about this kind of nice solutions, I mean, by graphical interface websites where you upload the data, you click several buttons. I think people start developing that, like, bioinformatics/data science is like one button solution. You click the button, something happens, you get some plots. And so that's good enough. So, but I think if you operate with the code, you really kind of feel the data. You really attach to the data, and you feel like you can experiment with the data a lot. So currently we rely on you guys, like to provide the biostatistics training, like for the students, and that's actually obligated for all the students to take the basic biostatistical class. But those classes may or may not to provide extensive code-based data analytics and not necessarily focus all the time on the hands-on, at least those basic classes which they are taking. So brings us to the point that we want to expose them to the code-based method and perhaps take the best of two worlds, like take some kind of fundamental biostatistic concepts that they absolutely need to know, but embed them with the hands-on. And I think there's a lot of opportunities for kind of like learning by doing, like you learn the concept, you take the data, you play with the data, write the code, you kind of feel the data, feel the concept, and go back and read again the concept. And I imagine it's more clear to you. So I think there's a lot of opportunities for kind of innovative teaching and hands-on things. Okay, so what we decided, we said, okay, it will be nice to have a class which specifically will be taking the data sets which pharmacists and pharmaceutical scientists can relate to, right? Something like they care about. If they run some command, it's not about just like cars or like some air pollution or something, but something that they can really relate and will be kind of excited to know those details from the data. So we said, okay, so, and we'll target the audience, I mean, the students from School of Pharmacy, which has no prior. I think it's safe to assume maybe 5% do have some coding skills, but the vast majority don't. And that was something that I personally do in my lab, the Python and Unix. So I decided, okay, I will teach the same. And we'll also focus on transparency and reproducibility, right? So, and again, I don't see technical barriers here, it's rather the culture, right? So can we kind of share that culture that every single code should be shared, all the data sets should be shared, publicly well organized, right? Not like dumping a mess on the GitHub, but rather like structure it. So can we create that cultures? So how we started? So I think right on the pandemics, I think hit it. So in the summer, we did it remotely. So we developed a very simple introduction to large-scale biomedical data analytic visualization class where we cover pandas, maybe seaborne for visualization, some bioinformatics a little bit, even though that was too much. And I realized how hard it is to teach bioinformatics because it's so overwhelming for them. But I think 100 individuals sign up. So we said, okay, there's interest, right? So, and feedback was overwhelming. Like people were really excited. They want to know those skills. They were happy to learn something. And we gave them individual projects. And I think in the end, whoever completed the project got the certificate. So there were like many people really work hard to analyze the data. And the data was simple as the structure, but it was large. We really wanted to discourage them to use any sort of Excel or anything like that. So we give them, I think, 1 million lines file, just to discourage them to use anything else. And then many people managed, many people who managed, and we got office hours. So people took it really seriously. So we said, okay, there is a demand, there is interest. And then in the fall 2021, I developed a class which I called Biomedical Data Science, or Introduction to Biomedical Data Science. And that was two units class, so now it's converted 4-unit class. And I think in the discussion, we want to give that as though, and okay, so in the difference that 2 units is just pure data science, just technical skill. As you can see, like Panda, Seaborn, some scripts, as well as some Unix, just because you need to know a little bit of Unix to run Git and the GitHub, like to run GitHub and connect to the, to run Git and connect to the GitHub and so on. But then we said, okay, can we extend to 4 units and cover some basic kind of biostatistics? You know, maybe at the ratio of 60% of the class will be biostats and the rest will be technical, but then they're very connected, meaning every concept you learn, you get hands-on. So this is where we are. And we have a discussion, can we provide that as the alternative as class for the biostatistics, you know, so people can take it if they want to go more into kind of hands-on. And I imagine that was actually the big discussion. Should we do it? Should we mandate code-based skills? And I think it was a mixed opinion. Some people say, you know what, we're living in the world of Python. My kid is coding Python. Everybody can do it. And some people say probably not. You know, will be some resistance because people didn't come to learn code in the School of Pharmacy. So I think it's a tricky question. I think it's similar to me if you think about the Excel, I don't know, 30 years ago, then it was developed. I'm sure it was not developed for all, in a sense, like I think they developed Excel as a very advanced tool that some can use. But now we're living under assumption everybody can do Excel. So I guess time will change, and at some point we'll be living under assumption that everybody can write 10 lines of code, right? So I guess it's a cultural change, but so far I see some resistance in the students. So I think it was a elective class, this one we did, but yet some students were just not getting it. Like, I don't know if they know about not putting enough effort, because we tried our best to provide TA. So it was always assistance available, office hours, so you can learn. And code is simple. We're talking about 5 lines, 6, you know, something extremely simple, even though, but it was resistant. No, I don't understand that. Okay, anyway, so this is what the goal is, right? So the goal is, you know, like them to be able, and emphasize again, large scale, right? We want them to operate large data, or at least know how to do it. Visualization, I think that's absolutely necessary discussion, how you communicate your results and so on. Most approach is statistical method, and this by statistical part. I guess, you know, it's becoming more popular when you do data availability statement and code availability statement as you submit your publication, and that's expected, how you organize your code and how you do version controls of your code and so on. Okay, so just to be on time, so just to wrap it up. So I guess there are a few ways kind of we thought about how to cultivate computational culture. So I guess you can do series of intensive hands-on workshop that serves some purpose. You can do new classes, new courses, which will be tailored to the needs of the students. Maybe you can do new concentration. That's something, you know, within the degrees which are traditionally kind of far away, or they stayed away from data science. But maybe it's time to create some tracks for the people interested in data science. New graduate programs and minors. I think minors for undergraduates or otherwise, you know, so that can be a mechanism. I think I will skip this. And this is the statement I was putting for a year already. So research is dedicated to all freedom-loving people around the world, to the people of Ukraine who fight for our freedom. And with that, thanks a lot. And we're looking for the students and the postdoc. Okay, thank you.
So we have time for a few quick questions.
I don't think there's anybody.
Oh, there isn't.
Doesn't look like it.
You usually have a class after here.
No, it's
Close to the end of the semester.
maybe it's there. It is not.
Okay, so we have time for some questions. You know, online or in the room.
Yeah, I have a question sort of relating to a question you had posed at the beginning of the talk about, you know, publishing these types of studies, which I do think are fantastic research. Like, absolutely, I want to know the results of benchmarking studies. But for a lot of these methods, these are sort of things that are continuously being developed or redeveloped or edited or things like that. So how do you think about benchmarking study sort of publishing at a specific time and being a snapshot of the methods that exist then, versus being an ongoing thing?
Yeah, that's a great point. And I think this is what it comes to kind of like, can we do continuous benchmark, right? Like that will be a ideal case scenario where it's like, you know, as the method keep developing, we also keep like tracking them, right? So yeah, so absolutely. And I think you're right, the snapshot, right? Like at the given moment of time, which given the publication process probably was a year or 2 years ago, right?
Yeah.
So this is what was, so you're looking at the past in a sense, right? So, but I think it's hard to incentivize, right? Like it's hard to, like what would be the incentive? You know, some incentive when you do benchmarking, you want to publish, right? And or like, this is like the incentive for the student. And actually to me, that's a great master student project for the master student, because it's very well defined, right? And, but it's doable in a sense, but, and also this is, to me, that's ideal project. And why I say that, because you don't care about the results. So this is rare. You know, we like, you know, when you develop a new method, you want that to be the best. Or if you want to do discovery, you don't want the results to be negative. But here, you actually don't care. And this is why actually what is called like Nature implemented, if you know that, like they implemented what is called a registered report. So basically the idea is that you provide the plan for the benchmarking and you send it to them with no result, just like what data you're gonna, and they actually kind of conditionally approve it or whatever, accept it. Just because it doesn't matter what the results will be, as soon as you stick to the comprehensive plan. It doesn't matter if all the tools are not working well, that's important to know. You know, if every single tool is nice and we solve the field, that's nice, you know, it's nice to know. So in that sense, but yeah, coming back to your continuous, yeah, I think it's like, it's a, yeah, that will be kind of, it's a kind of interesting question how to do it, right? And then, you know, that's actually, we need like more people in the field like to come up with the kind of creative, innovative solution for that.
I mean, I think like, get your opinion on it, because so in statistics, we create a new method, we do our internal benchmarking, which is simulation. Often the problem is we simulate the data that comes from the model that we created, right? And so then our model does the best for our situation, and then we compare it to other approaches that would be different, and they compare it to their approach. And then also the other thing is the mentality of the method has to be the best. And in statistics, that's often power, right? But that's not always necessarily the goal, because you have performance like other issues and simplicity, interpretation. Like, so, I mean, it really goes back to the same question of creating this culture, because if benchmarking studies and or every individual method creates the data, you know, it would be really nice if I created a new method, but I could simulate under my model, grab the data from another paper that was just published, and you know, and be able to pull all these sources of data and do benchmarking with that,
Yeah.
right?
No, I think ideal, if you think about kind of ideal case scenario, probably we need to kind of blind benchmark, right? Like there is some kind of server or whatever it's called, right? And then basically the data, you don't know what is the data. And you submit your tool, they run it and give you the results, right? But it's kind of like, hopefully it's comprehensive as you said, across the papers, so on, and using kind of the metrics we agree. But you cannot overfit, you cannot train on the data, you cannot tweak, so you get the results, but you don't know what is the data, and every time maybe the data is slightly different, like some, so you cannot like tweak it.
But the problem is that, no method, is often universally best.
Yeah.
There's give and takes.
Oh,
right?
yeah.
You design a method because we think it'll work best with rare variants,
Mhm.
more common, or this or that, and so in doing benchmarks across the whole range of variation, there needs to be a nice
Yeah.
summary on when to use this, when to, you gave it there, like when you think you have, what was it, T cell,
Mhm.
then use this approach, or
Yeah.
don't, right?
Right.
That's a simple solution.
Right.
It can get more complicated.
Right. No, but I think it's like, I think that's another thing we need to change, exactly as you're saying, not every method is the best. It cannot be true. So now we are talking about different scenarios, different data, or different metrics, right? Some has the nice power, but it's not interpretable or not scalable, right? And so I think I didn't put it here, but we put like as this Pareto frontier, like so there are few, like parameters, like for example, you want to run the data across the very large study and you want to interpret the results, right? So that's your primary objective, right? And then you want to be, decently accurate, right? So I think then this is one method, but if you only care about accuracy and you don't plan to run in the large data, probably will be another method. And I think that's kind of, communicate, if we kind of can communicate that, we can kind of remove a little bit the tension of like, oh, who is the best, am I the best? Like, like the tension of like ranking. It's not kind of ranking then. It's just saying what are the methods good for this kind of problem and for that problem or for those results and so on, right? So it's more complex, just the ranking.
Any other questions, thoughts? We will be continuing out like for lunch afterwards this session, so if people want to join us. Yeah, if not, thank you all for joining.